I'm Dr. Jereme Spiers and I'm a postdoctoral research fellow here at the John Curtin School for Medical Research. The brain itself is a fascinating thing. We're trying to understand different puzzles that are happening in the brain. One of the ways that we can begin to look at this puzzle is through some of the factors that we know change early in Alzheimer's disease. And one of these is neuroinflammation. This is typically caused by an activation of the immune cells within the brain. Now, they begin to produce a number of different proteins and other molecular factors that you would typically see in an infection. And in Alzheimer's disease, this actually happens quite early. One of the challenges that we have now is to try and understand what's causing this neuroinflammation. One of the problems that we have is that we don't really understand the root cause of Alzheimer's disease. We have really excellent pathological markers that we have in proteins like amyloid beta and tau, and we know that these are involved in the pathology. But what we don't really understand is the process that makes those toxic very early on in the disease. For my project, we'll be looking at a specific kind of cellular communication that we know gets changed during inflammation in Alzheimer's disease. So because we know that immune cells are incredibly important in developing neuroinflammation and as well as in Alzheimer's disease, we really need to know what those immune cells are saying to each other. And there's a few different ways that we can look at this. One of the ways by looking at cellular communication directly, so cell-to-cell communication. So understanding how cell-to-cell communication is being impacted by things like neuroinflammation in Alzheimer's disease is critically important for us to develop new therapeutics. And to do this, we look at things called extracellular vesicles. And these are small lipid membrane particles, so just small packets of information that cells send out to each other in order to talk. What we're starting to see is that the proteins that are being sent out by immune cells are actually saying a lot more than what we originally thought. And the way that they do this is by being packaged within these extracellular vesicles and then communicated to different cells. If you imagine the packets of information that cells use to communicate with each other are something like M&Ms, there's a number of different colours of M&Ms, and these might represent the different cell types that those packages are coming from. Now, normally when we isolate these, they come all together and we can't actually identify what an individual cell type is saying to one another. So what's different about this project is that we're going to be able to isolate the specific packets of information which might be representing, say, the red M&Ms that come from the immune cells of the brain, and we're going to be looking at exactly what those cell types are saying to each other. The funding from Royce Simmons Foundation and Dementia Australia Research Foundation has given us access to some of the most sensitive equipment in the world for looking at these particular packets of information that we just haven't been able to see before. So understanding one more piece of the puzzle of this really complex problem is critically important for us to develop new therapeutics going forward.